All right, so welcome everyone. We're going to go ahead and get started. I want to welcome you to the North American Food Explorers 2023 virtual conference. My name is Chris Heater, and I am the president of NAFEX, as we call the organization for short. I will be serving as the facilitator of the session, and we are here talking about the soil food web today. Before I get into Dr. Elaine's bio and information about her, let me just very quickly give you a few housekeeping items so that we're all on the same page. First of all, this is a webinar, so unlike the Zoom meeting that we just had with our annual meeting, your audio and video features are automatically disabled. You'll notice that you can't really chat with each other like you would in a Zoom meeting, so keep that in mind. Second, we are asking you to put your questions in the Q&A tab, which is at the bottom of your screen, as opposed to the chat. Only use the chat if you have a technical problem. We've got people monitoring both the chat and the Q&A, but we'd like all questions to go to the Q&A so that we can scroll through those and watch those and feed those back to Dr. Elaine as we go. And finally, this webinar is being recorded. It will be made available to all of you, and after the conference, we've got a team of volunteers working to make that happen. They should be ready early next week. They'll be available on nafex.org. When you log into the website, you're going to log in with your email address, and you'll be able to access all the recorded sessions and any bonus resources, anything that the presenters have provided as part of their talks. Just a few words about NAFEX. So we know many of you just joined us this week for the first time. NAFEX was founded in 1967. The North American Food Explorers is a network of individuals throughout the US and Canada. It was actually started by two gentlemen, one from the US and one from Canada. It was a joint effort when they began NAFEX. And it's devoted to the discovery, the cultivation, and appreciation of superior varieties of fruits and nuts. NAFEX members share ideas, information, experiences, and fruit propagating material via our website, our social media channels, fruit-specific interest group meetings that we have, and our annual conferences like this one. In the past, many of those were in person, but of course with COVID now, much of this is online. As a paid NAFEX member, you get 4 editions of our quarterly Pomona Journal each year. That's available both digitally and printed, depending on your membership choice that you chose when you signed up. And you have the ability online to search 50 years of the Pomona in our digital library. You can search that by both year and keywords. There's just a wealth of information in those archives, so I highly encourage you to spend some time going through those when you've got that chance. And if you haven't already, please visit our website at nafex.org. Once you log in, again, as a member using your email address, you're going to find so much stuff available to you as a member when you log in. So when you've got a rainy day, just take some time and you can check all of that out. So with that, I want to pivot to introducing our speaker. Dr. Elaine Ingham is our speaker today, and she's going to be talking about the soil food web. She has advanced the knowledge about the soil food web for more than 4 decades. She's widely recognized as the world's foremost soil biologist. She's passionate about empowering ordinary people to bring the soils into their community back to life. Dr. Elaine's soil food web approach has been used to successfully restore the ecological function of soils in 6 continents. In the mid-1980s and 90s, after earning her PhD in microbiology, she served as associate professor teaching forest science, botany, and plant pathology at Oregon State University. She later founded Soil Food Web Incorporated, a consulting firm and training program dedicated to advancing sustainable agriculture in which a healthy soil food web promotes plant growth, reduces resilience on inorganic chemicals, lowers farming input costs, and regenerates ecosystems. Her firm works towards its mission through direct consultation, workshops, online training, and public speaking engagements, with a principal focus in North America, Latin America, and Europe. Through much of the last decade, Dr. Ingham has also applied her knowledge to support allied organizations. She's the author of the USDA's Soil Biology Primer and also served as the chief scientist for the Rodale Institute and managing scientist at Environment Celebration Institute. She consults throughout the world, but calls Corvallis, Oregon, her home. She offers training classes and certificates at thesoilfoodweb.com, and I know several of our members have been taking or are taking those courses. So with that, I would love to turn this over now to Dr. Elaine. Thank you so much for being willing to speak to us today, and we look forward to your presentation.
All right. Yeah, it's my favorite topic. So look out, I'll probably go more than an hour, or you're going to have to cut me off. And we can just play that by ear. I like to start out with this picture of me in an agricultural field. Yes, I do actually get my fingies in there and take soil samples on a fairly regular basis. But I like this picture because it shows us that we can grasp a great deal of what's going on with the biology in this soil just by looking at various factors. Most people would dismiss those things as, they're just, that's the way they are. But they're not. They are messages from Mother Nature about what's going on in your soil. And we need to learn to interpret this information so you know what the correct fix to prevent all of the problems with the things I talk about. Pictures like this, I know that we have anaerobic areas in the soil, so diseases are going to be a huge problem here. We're going to have no way to hold on to the nutrients if you put inorganic fertilizers into this. Well, it's not really a soil. I think of it as dirt. That's what it is. It can't hold the nutrients. There's nothing there to hold it. It is the biology in your soil that holds on to those nutrients, that holds the water in place. Yes, you've got to be below the evaporative level for water to be held, but you also have to have the microorganisms that build the aggregates, all those different sizes of aggregates that exist in soil. You don't have them in dirt. Take a look to the left-hand side of this picture, and you can see how this dirt has been driven to the point of not being able to do anything other than be a physical place where you stick the roots in and cross your fingers and hope that it's going to grow. When we look at that, it's been tilled and tilled and tilled, and every time you go over, when you till your soil, hopefully you're starting out with soil. So you till that soil, you have killed 50% of the beneficial organisms, the bacteria and the fungi, protozoa, nematodes, microarthropods, earthworms, enchytraeids, all of those critters, their populations have been reduced by at least 50%. Okay, they didn't stop at one tillage, because look at the right-hand side of that picture, and you'll see these clods of compacted material, just rock solid. You jump on them and you can't get them to break open. So they run the tillage equipment over it at least two times more than what you're seeing here. Well, so you lost 50% of the really important microorganisms doing a lot of work for your plant. With that first tillage, 50% is gone. The next tillage that you can use going over this, you're going to lose another 50%. So here we are at 75% loss. And then they're going to till again. And then they'll till again if it hasn't turned into the sand, silt, and clay. And so there is no biology left, and you have to apply inorganic fertilizers and pesticides and all of those toxic chemicals that are destroying our waterways. So we have to understand what we're doing and what's going on here. So there we go. Just, biology is the ultimate tool. Mother Nature has been using the biology ever since the very beginning. How long have the bacteria been on our planet? 4.5 billion years. Fungi came along 3.5 billion years ago. Each of the different sets of organisms in the soil that build these intricate loops and intricate interactions. And every time we improve that biology in the soil, we can increase the levels of fertility and production. I don't usually like to tell people exactly how far we can take this, but it's far it's far better than what you get by using inorganic fertilizers and pesticides and tillage that just is not going to do the job for us. We increase fertility and production in some places as much as 50 to 100% of the time. As you go into the second or third year of using biology instead of toxic chemicals, we can increase yields up to 1,000-fold. Feeding the world? We are. Not the chemicals. That's not going to do the trick because those chemicals kill these organisms. So my, Bachelor of Arts, Master's degree, PhD, if you want to email me, ERI, those are my initials, at soilfoodwebinc.com. Also, I have an email, soilfoodweb.com for the school where we teach people, more in depth, where you learn at your own pace, and that makes it pretty easy to, there we go, makes it pretty easy to go at the speed, learn as you need, and not as, somebody pushing you and saying, well, the test is Friday and you're going to fail it or you're not. So this is our team at Soil Food Web Inc. Incorporated. So hopefully you'll come to know the folks in the in SFI. If you want to know what's going on in the soil, we can do that, teach you, train you to learn about, be able to properly predict balances between these organisms. So if you look at the sun coming up over there on the left-hand side again, how do you store the energy tied up in sunlight? And that must have been a fun job to figure out if you were Mother Nature. How does that energy get stored? Well, in photosynthesis. And of course, that means you've got long carbon chains. If you don't use that stored energy, you're going to get longer and longer carbon chains, but you're not going to be able to, at a certain point, you're not going to be able to grow anything more above ground, because you've got to get the nutrients from the organisms in the soil. So this whole process of growing crops, you have to realize that, yep, sunlight is giving you the photosynthesis, but then those roots have to go down into the ground. And the nutrients that your plant requires for everything else has to come from where? So when we're looking at these, I'm pointing at them over here, and I realize that I don't have my my arrow out. So over on the left-hand side, you can look at all of those roots. How do those plants get the nutrients that it requires? How do algae get the nutrients that they require? Well, with the plants, we're going to have exudates being produced, put out into the soil in order to message the bacteria and the fungi. And if you look at the second trophic level, you can see where the bacteria and the fungi sit. Well, you can also see what the bad guy nematodes, the root-feeding nematodes that can harm and kill plants if you don't deal with them properly. Well, we'll get around to dealing with them properly in just a few minutes. So the bacteria and fungi are the good guys, presumably. As those plants are growing, they're doing specific things to suppress the diseases and the problem organisms using the whole food web here. So bacteria and fungi are getting messages from Mother Nature in the form of proteins and different kinds of amino acids to tell the fungi and the bacteria which nutrient and how much does the plant actually need. So the bacteria are going to go forth then, because they'll get more food if they go back to the plant and say, here you go. So the fungi and the bacteria out looking for nutrients. Typically, they're going to end up at the edge of a mineral particle in the soil, and they're going to use their enzymes. The bacteria and fungi use their enzymes in order to pull the nutrients that the plant wants out of the bilayer of silica. That is what your plants, which is what stores all the nutrients ultimately in the parent material. So the bacteria and fungi pull those nutrients out. That's what the plant requires. The fungi and the bacteria are in that root system, so they're hanging out, waiting for their turn to be called to give these nutrients to the plant. Well, how does that really work?
Dr. Elaine, can I interrupt for just one moment? I want to make sure that we're still on the soil food web slide. And if you would just say next in between each one, and I'll make sure I'm coursing along with you. Are we still on the soil food web slide?
We're still on the soil food web slide.
Awesome.
So look in the middle of this page, and you're looking at protozoa and nematodes, fungal and bacterial feeding nematodes. So what do they eat? I think you can figure that out. Microarthropods, the top layer of this third trophic level. Microarthropods are also given information what the plant requires, and the microarthropods will go around searching for those things. Eventually, they're going to move into the root system, and they find the bacteria and fungi that they want, and they will consume those bacteria and fungi because the nutrients of the mineral nutrients that your plant wants is in such a higher level of concentration in the fungi and the bacteria than the things that eat them, the protozoa, the nematodes, the microarthropods. Because of that discrepancy, when the bacteria and fungi get eaten by the protozoa, nematodes, and microarthropods, large amounts of the nutrients that the plant was asking for are going to be released because the concentration of nutrients in bacteria and fungi are so much higher than they are in any other organism that you see on this page. And so that excess is released right around the root system, and the plant immediately takes up what it needs. When it has enough of boron or enough zinc or enough nitrogen or phosphorus or sulfur or magnesium or calcium, any one of those things, the plant takes up what it needs, and then it puts out an exudate that tells the bacteria and fungi, here's what else I need. So this is an ongoing cycling process. It never stops. Even in frozen soils, you'll find some percent of these organisms that are still functioning, those things that like it cold. So Mother Nature has just the most amazing abundance of how do you get nutrients released for the plants to be moving along. So we go to higher levels of the trophic system, macroarthropods, predatory nematodes, which are much larger than most of the other nematodes. They're eaten by birds and other animals. And of course, it's human beings that are, we think, at the top of this food web. So there's so many other things I can go into here and talk about, but I want to go ahead to the next slide and cover a few other. Let's see, and I don't get that. I have to. Yeah, I've got a different set of slides than you do, so this could be really interesting. At least I found them. Okay, so a lot of times people will use the incorrect terms. So I want to make certain that you all understand that dirt and soil are two very different things. So dirt is basically just the mineral nutrients that are inside the sand, silt, and clay. And the bacteria and fungi will go and pull those nutrients out of those silica bilayers. But that dirt does not contain any life. And so if you go down deep enough in your mineral materials, if you have toxic chemicals that are being applied to the surfaces of your soil, you're very quickly going to turn them into dirt. So mineral nutrients inside that, those nutrients are not plant available. Your plant can't get those nutrients out of these mineral materials. That's beyond the level that that they can obtain anything, pull anything out. So you have to have soil. What's the difference? Well, there's the mineral component of the soil, still just like we had in dirt, but in order to get those nutrients, we've got to have organic matter to feed the organisms in your soil food web, and you've got to have the organisms themselves. We have to have the predators of the bacteria and fungi, protozoa, nematodes, all those other things, food loops that you go through, and understand exactly which combinations of organic matter and organisms and how are you going to feed them and who gets what. We've got to have the predators in the system. This whole food web has to be there, or you cannot obtain the nutrients that your plants require. So when you look at where do all the important factors come from in soil, you go through photosynthesis, but everything else, the only thing your plant is getting from above ground is that photosynthetic process. All the nutrients come from the soil, and you have to have the organisms in the soil. So hopefully I've beat on that one enough. And next slide, if you would. Okay, so when I initially worked with soils people, people in agronomy or any one of the departments related to growing plants, they always were after the soil texture as being that's the determinant of whether you've got healthy soil or not. And that's just absolutely not the case. Clay, sand, silt, loam, these are purely minerals. You have to have the organisms in your soil to pull these nutrients out, to build structure within the soil, to be able to build the soil itself. You've got to have not only the soil, sand, silt, and clay, but you've got to have the organisms as well. So next slide, please. We want to know how these functions occur in the soil. And if all you say is that these things happen in the soil and you don't know how, you're going to misuse them. Every time we apply inorganic fertilizers to the soil, we kill the organisms that are not tolerant of that level of salt. Most inorganic fertilizers are salts. That means they're pulling water away from your plant and away from the microorganisms. Your plants can deal with a lot more loss of water than the bacteria and fungi can. Protozoa, the nematodes, the microarthropods. And so use of inorganic fertilizers are going to wipe out the very organisms that would have otherwise pulled those nutrients from a place that Mother Nature has been storing them for exactly that thing to happen. So we have to understand how the functions occur and who does those functions so that we can work with nature and not against her. And unfortunately, we have, for the whole history of agriculture with human beings, from the very beginning, we have been destroying the soil and turning it back into dirt in our agricultural efforts. We have to stop this. And I don't care whether we're dealing with trees or we're dealing with shrubs or we're dealing with, weeds. The green revolution has failed because the organisms that do the work were killed by the practices recommended by the green revolution. How dare they call it the green revolution? You have to have the entire food web for your plants to be healthy, for the soil to be healthy. Next slide. So some of the things we want to monitor, we want to check and see if the biology is present. So we want to be counting, determining the beneficial organisms, and make certain that they're at the correct levels. We want to maintain those levels and the balances. We want to know if we've got compaction. Why is compaction so bad? Well, compaction is the compressing of the surface of the soil or any part of the soil at all, and preventing oxygen from being able to move into that area. If you've got very little oxygen in an area, you're going to have anaerobic microorganisms starting to grow, and the anaerobic organisms are deadly to most of our beneficial organisms out in the soil. So all of a sudden you stop holding water. You have water that puddles at the surface of the soil, and then when enough water accumulates, it was going to run downhill to whatever river, lake, stream, and of course, then that goes out, ever increasing the concentration of toxic materials, because that's what we're putting on our soils. So we want to make certain that the watering requirements are correct. Let's not water too much. Let's not water too little. But how do you really hold on to water in the soil? If you don't have biology, that water is going to go right through and end up in your rivers, lakes, and streams. But if we can get the bacteria and fungi working on making microaggregates, so we're starting to tie things up in the structure of that organic material in the soil. A little bit higher level, fungi and protozoa, nematodes are going to be building the superhighways where oxygen can move very rapidly from the surface of the soil down as deep as the root goes. How far down does the root of the plant go? Well, it's real important for yields. So we want to be looking at, how deep do those roots go? If we till the soil, where's the cutoff that we're destroying that biology? And what happens to all of those root systems that are now disconnected from their source of food? So we want to understand how all of these interactions go, and compaction is a hugely important factor here. If we have compacted soils, oxygen can't move through, you are mostly promoting for the diseases in the pest species. You are reducing fertility. So these are all things you can use to measure the health of the soil. And of course, with the soil food web, we want to look at the biology. We want to know what's going on with the weeds. Weeds are promoted in early successional stages, so that, we hate weeds. If we would just move the biology along a little faster, you can avoid the weed stage of life altogether. It's not something we should be having this much trouble with, but you have to understand how the soil works. Sequestered carbon. It always surprises me when people say bacteria are the only thing that sequester carbon. Wrong. It's sequestered by both bacteria and fungi. And fungi, certain sets of fungal species, are much better at tying down carbon and holding it within their bodies. If you look at a fungal hypha growing as, it's like a long tube, basically. When the plant is working on breaking down organic material, it's going to take any excess carbon and put it on the inside of the fungal hypha. And there were people writing about this kind of situation happening back in the 1950s. And somehow it's just nobody cares. No one understands that biology is important. And well, yep, I guess so. When I was a graduate student trying to decide what I was going to look at for my PhD work, I was really interested in the fungi and really wanted to understand saprophytic fungi more than mycorrhizal or other things. And so my major professor and I worked out a proposal, and then my major professor, Dr. Don Klein, said to me, go and talk to all of the people in the agricultural side of things. So I made appointments to go do that and sat down with every one of those folks and asked, is my PhD project of interest? Am I going to get a job once I get finished with my PhD? And so they read it over and just kind of slam the front page down and say, you'll never get a job with this. When you think about microorganisms in the soil, there aren't any in the soil. And I was, oh, really? They don't do anything. Bacteria in the soil doesn't do anything. It doesn't help your plant at all, period, full stop. Well, of course, if you're killing everything in the soil that looks like a bacterium or a fungus or whatever, of course you're not going to ascribe any benefit because they've all been killed. What happens when you don't kill them? Well, we've shown that there's way more carbon being sequestered in the biomass of the organisms, but you have to have the organisms first. Okay, beat on that for enough. Next slide. And I think, yeah, I'm still, okay, next slide. So when we're looking at, and I think we're gonna get this overlapped. So when we're looking at how deep do roots actually go, this is a plant from the golf course. And we took those seeds and we applied them in areas that were completely tilled, have lots and lots of compaction, and compared it to an area where we put the proper biology into the soil. We didn't even till it. We just put the biology into the surface as compost. And yes, you have to be very careful to make certain that you're using good compost. I have killed more plants by putting around their root system something that somebody said was really good compost. And I managed to kill all of the plants. No, it wasn't me that was at fault. It was the lack of beneficial organisms. The compost they made was just one horrific, anaerobic, disgusting smelling, horrible smell, and it killed all the plants. Here we took up compost that had all of the organisms that this plant needed to be able to thrive. And this is in a golf course. We checked a couple times during the growing season to make sure everything was a-okay with the biology in the soil. We looked at how far down the roots seemed to go. So at the end of the growing season, just before things started to get pretty cold up in Seattle, Washington, we took a 10 inch wide, no, a 10 centimeter wide pipe and pounded it into the ground, and then we had to get a great big tractor to pull that out. We cut the plastic piping from around that root system, and that's what you're looking at here. This was cut. This was cut grass 3 days before this picture was taken. So when this plant was being mowed, did all of its root system fall off? And today people will laugh at the idea that anyone could have thought that was the case with grasses or other things. But because of the chemical training people have been put through, they really, truly were adamant initially that the root systems only go down 2 or 3 inches and then they go sideways. Well, yeah, if you're a city street tree and you're been put down on concrete, good luck. You're not going to have the organisms that you need. You're not going to have those root systems growing down. So with this, that root system was at 4.5 feet down into the soil, and we had done nothing but put biology on the surface. And the organisms built the structure all the way down. So oxygen and water and organisms could all do their proper thing at the proper time. And that particular golf course area in the golf course was absolutely the best. And of course, they started using biology because it's a lot easier to put down 1 or 2 applications. And then that's it for the biology in that system, for needing to put on more biology. So save you a lot of money. Things are more beautiful because everything's vibrantly growing. So next slide. Let me know when you're going to cut me off. Okay, so the soil food web, hopefully you understand a great deal more. Keep going. I think I have a few more. Yep, I'm going to have to do this. Yep. So soil microbe interactions make nutrients available. Does anybody want to contest that that's an important thing to do? I've talked to you about the predator-prey interactions. Well, of course, mycorrhizal fungi are told by the plant that the plant needs X, Y, or Z, mostly phosphorus, some nitrogen, potassium, things like that. But that the plant tells the mycorrhizal fungus to go out and collect this much more of the nutrient. The mycorrhizal fungus brings that food, those nutrients back to the root system, and the plant and the fungus exchange how much each of them is going to get out of this deal. Rhizophagy is one that's just come on the view of people, where at the root tips of certain plants, the root puts out food for the bacteria and fungi, and so they're having a party at the tip of the root of that root, and the plant, the root, will eliminate the cell wall between the outside and the inside of that root system, so that all these bacteria and fungi, protozoa, maybe even a nematode or two, move inside of the root system, and then the plant shuts them down. They walked into the trap. And so the plant puts out an oxidizing agent that eliminates most of the cell wall of the bacteria and fungi, protozoa, and nematodes. So all of these nutrients being pulled out of those organisms, out of the bacteria, fungi, protozoa. And at a certain point, I think it's kind of fun to watch. All of the bacteria and fungi, etc., decide to exit the scene because this is getting pretty destructive on their cell wall. So they move up and leave through a hole or through some mechanism, we don't know what, and the microorganisms move back out into the soil. And then they move their way down towards the tip of the plant, and start to grow, start to do the things they were doing previously. And then the plant invites them in. Now they're in the trap, and then they escape, and it just goes over and over and over again. It's like watching somebody who loves, to ride on the scariest ride in that they can think about. So, are there more ways? As we go on, will we be discovering more ways that plants interact with the microorganisms in the soil? But remember, You gotta leave the organisms in the soil if you want to see their benefit. So, next slide. Flavor, of course, is determined by nutrient balances. So when you're looking at your trees, when you've got an orchard of something, you've got shrubs of some kind, uh, the flavor is so often determined by the concentration of the nutrients and the particular compounds that can be made given those balances. So it's really important to have these microorganisms feeding your plant, and your plant can get picky about exactly what it needs. So a very important part of what happens early in the growth cycle of this year's crop. At a certain point, we always see that the plant stops taking things up. They've got all the nutrients they need in their bodies, and now they can grow some of the most luscious foods that you want to talk about. And it's a really different understanding of how all of this works. It's not right at the end, your plant is struggling to, pull whatever it can so it can finish seeding. We should have all of that process done and finished halfway through. And so a lot of time to mature. Nutrients need to be captured and held in the soil. We don't want our nutrients, nitrogen, the nitrates, the ammonia, the phosphates, the sulfates, all of those things. We want them held. We don't want to have to put in another load come springtime. Would it save your budget any if you didn't have to go out and apply? That all of these nutrients are in a cyclical process, and we don't need to be losing all of our nutrients and causing all the destruction downriver. So we want to stop runoff. Well, what's causing the runoff? You have compacted soils, and the water can't infiltrate into the soil. They're, going over the surface of your field. Leaching is occurring. So we've got to get the biology back in the system so they build the structure that will hold. I've worked with wine grapes and table grapes in Australia for a number of years, and we've seen as soon as we get the biology in the soil, we can hold all of the water that an entire crop of those grapes is held in the soil, and so you don't have to be putting in giant size reservoirs that you could better use growing trees in. So next slide, please. Okay. We suppress diseases because we have those organisms that compete with, inhibit, that means they're making antibiotics basically, but in very small amounts, just in the place that it's important. As human beings, we make the whole entire 1,000-acre plot be treated with toxic to the microorganisms materials. And so what else are you destroying when you're trying to just inhibit the disease-causing organisms? We have situations where there are specific bacteria or fungi or protozoa or nematodes or microarthropods that go after the disease-causing problems. So through the process of consumption, there is no reason to be worried about diseases. No more pesticides are needed. Thank you. We're done with that. And so, I've been invited to work on some of the hardest problem fungi, for example, and we're showing complete control without using any toxic pesticide. We want to suppress weeds by balancing nitrate and ammonium. Nitrate promotes the growth of the weeds. Ammonium promotes the growth of the plants that require more fungi than bacteria. So we can control this to some extent. I also want to get through that stage where the what's through succession, you're getting mostly weeds because you're at that ratio of fungi, of nitrate and ammonium, fungi and bacteria. So I want to move on through that successional stage in like a day or two instead of a couple of years. How much less is it going to cost you to have taken care of these particular problems? Let's use these microorganisms to decompose the toxins. They've got enzymes that are, the enzymes made by the microorganisms. Let's clean up the mess we've made and not fight nature. Let's work with nature. Next slide. So building soil structure, I've talked about micro and macro aggregates before, how we fill up 50% of the pore space in these aggregates. And so you don't have to have outside reservoirs. Your reservoir of water is right below that evaporative line. And you need to have the biology to be able to build these aggregates. And again, examples through most of the places in the world that have drought problems, they start working with us and they don't have to use the amount of water, or sometimes they don't have to use any extraneous water. It's all stored in the soil as deep as you possibly can get with your root system. How far down can root systems of plants go? Well, I showed you that example at 4.5 feet. How about under a sequoia or under mature Douglas fir? I know the mature Douglas fir trees can go, their root systems can go down 250 feet. Other people looking to kind of say, Elaine doesn't know what she's talking about. Turned out that they were working with some cedars, if I remember correctly, and they were getting root systems down to 300 feet. So it's not just us. Everybody, I always think of poor city trees. Their life is being cut off at an early stage because we don't understand about soil. We sequester carbon, and this is the C-Dan ratio of bacteria versus the carbon and nitrogen ratio. Sorry, carbon to nitrogen ratio of fungi. Fungi store much, much more carbon in their hyphae than bacteria do. And so it's sad when, especially in forested systems where we're dealing with orchards, that people push bacteria. It's harming your plants. You've got to get the balance correct. So there it was. Succession. When we start out after a catastrophic damage being done, after a wildfire goes through and the temperature in the base of the root system have reached 600 degrees and nothing is left. We want as rapidly as possible get biology back into that system. Maybe it's moss, maybe it's something pretty simple, or it's the right kinds of plant material trying to grow. So early successional grass species, some of the herbs, some of the spices, onions of certain type would be chosen for in here. And look at how much has changed with respect to the bacteria down at the bottom underneath the picture of the ecosystems going to the right. You can see there's a space that says bacteria 10 micrograms, fungi at 0 micrograms. That's for where you're not growing plants at all. That's, if that's what's in your soil, oops, not soil. It's dirt with the worst of ways. Here, when we're looking at more of a balanced situation, bacteria 500 micrograms, fungi 250 micrograms, you're setting the stage for most of our veggies, for the salads and Brussels sprouts and things like that. But as these organisms, as the bacteria and fungi work to help grow that plant better, the plant is going to start pushing, well, actually the bacteria and fungi first that start to push for other kinds of crops growing in here. So it's really the bacteria and fungi that push the whole process of succession. Well, once you've got the bacteria and fungi in here, now it's really easy to grow tomatoes or strawberries or those kinds of plants. Moving over to where these are shrubs now growing, the classic plant succession. Well, but it's preceded by a bacterial, fungi, protozoan, nematodes, microarthropod succession first. That's got to happen first, not last. So here we are at our fungal to bacterial ratio, 600 micrograms each. As the plants start to move into the larger shrubs, into the earlier deciduous part of the of plant succession going on here, you'll see that now we've got 800 micrograms of fungi, 500 micrograms of bacteria. And so we've gone fungal dominated. And that's what any of these tree species, deciduous early, my experience is early in succession. It's mostly deciduous. As you grow older and older forests, they're going to become much more conifer dominated. Well, so 7,000 micrograms of fungal biomass to 700 micrograms of fungal biomass. And I think there's one more slide. Yep. Just to point out that down here at this end of succession, nitrate is typically the predominant form of nitrogen. As you move into mid-successional areas, the ammonium starts to play a much more important part. And then as you go on through into all conifers, it's typically pure ammonium. And so we haven't had a chance to go everywhere in the world. So it would be nice if we could tie up these observations and see how much of the world is run by these kinds of successional processes. Are there other successional processes we should be looking at and understanding? Well, we've got trees, and a lot of those trees have had very few, if any, people looking at that process. So I think I will finish here.
Fantastic. Thank you. All right, we've got some questions. And so I'm going to start. Where shall I start? So I want to, Pat Holland brings up toxic metals, and I was interested in this as well. So you've talked about toxins, but toxic metals are more of they're down to their final element. So how do, soils that are heavily contaminated with a toxic metal, how do the bacteria and fungi deal with that?
Bacteria and fungi are pretty good at being able to sequester those toxic compounds, kind of, and I usually look at things at this, level, and you're talking about something that's down at this level, very fine-tuned, and you have to have the right microorganisms that are going to be able to grab those heavy metals and put them into a structure that means that nothing else is going to get a strong enough dose at any time in the near future. Now, what happens when something's been in that nice aggregate, happily doing its thing in there and not really harming anything? Does it just kind of sooner or later disintegrate because the concentration is so small that it doesn't matter? So we've got to get some materials that are in those stages or know that they were in that kind of situation. So we've got a bit more work to do at that end of dealing with heavy metals.
Yeah. I've heard situations where people have recommended like sunflowers. Sunflowers will take up some heavy metals, but then if you just chop and drop those, you're still keeping those heavy metals right there, right?
Yeah. And I don't think we can change the structure of a heavy metal and make it a different heavy metal. I don't think we figured that one out yet. And then what do we do? What happens when we discover that, oh, that was an important nutrient for somebody? So a long way to go when we're dealing with that subject of heavy metals. But we do know that, if you're in a Chernobyl kind of situation, get out there, get the plants into the system, get the microorganisms. You've got to have really good compost, and then you'll watch the heavy metal be reduced in the food, and
Right.
then they haven't cared to figure out where all the other where did it all go? What did you do with it? On whose door did you plant this one? So there's some interesting things going on out there, and I'd like to. Oh, it would be fun.
Right. Let's see. So you've kind of answered this, but let's review it because this was a question early on. Caitlin asks, would you recommend low till system for annual vegetable production?
I basically say no tillage at all. Why do you need to till? If the biology in your soil is exactly what your plant wants, if you go out there and till, uh, you're going to cause yourself problems. Now, let's differentiate between the term till and the term of a light, me and my, I love it when my, yeah, so, you're going out and you're with the, yeah.
Like a broad fork, maybe?
Yeah, with a broad fork would be, or just to smooth out the surface of the beds if that concerns you. And I always like to put in cover plants. I don't want any drops of water coming from the heaven because, how high up was that drop released from that cloud? And so what's the energy impact of that drop of water coming down and harming everything because it's a compacting, it's just as bad as driving your trucks over that surface. We want to have cover plants. So as that drop comes down and hits the top side of that leaf, the leaf bends, it hits another leaf, that leaf bends, and most of the energy from that fall, 5 miles, maybe 10, it is not going to have the harm to your soil. If you don't have anything, taken up that impact, you're going to be compacting all of your land, and then you're going to have to put out another application of the compost extract to be able to neutralize that effect of something smacking into your ground quite so hard. So I think that's probably it's a bigger problem that nobody's looked at. I haven't had the ability to do that work, but yeah, send money to my funding Soil Food Web Incorporated, and we'll do that for you.
All right, let's see. Janet asks or says and asks, I live in an area that gets some areas of puddling, apparently because of an underlying peat soil. Are peat soils redeemable?
Oh, yeah, they are great organic material, and I probably wouldn't blame, place the blame on the peat layer, because the ease of dealing with it is just to take a pole, punch it into the ground, go through your whatever layer it is that you're dealing with, make sure that that goes all the way through, and now fill up that you know what you just left behind when you pulled rod back out, there's a nice open all the way down to as deep as you went. And now you're inoculating all these wonderful organisms to do their job of building structure. So next year, you won't have a problem. So try to get off of that. We got to till it up. We've got to disturb it. We've got to make it all light and fluffy. Do you know how rapidly light and fluffy is compacted? It only takes one rain event. Only one time of doing, watering your property. So choose the right path.
So John asks, and a couple of people have commented. So John says, how do we get nutrients that have been leached deeply into the subsoil to become available to our plants? If using plants for this, are they deep rooted enough? And someone mentioned comfrey and other bioaccumulators.
Yeah. The easiest way to get those, chase after those nutrients that are, slowly moving, they were beyond the level of the root system of the plant you were growing. And it was probably compacted there, so it's much more capable of growing deeper and deeper down if we would just understand what we're doing. So again, punching your stick and filling up with compost extract, or plant trees just downhill, or whatever way the water is moving through your systems. Plant some trees whose root systems are going to go down 250 feet, 300 and something feet. So you could even do a gradient where you've got, here's all your vegetables, and then here's a grassy area so you can have fun with your kids. And then the shrubs, and then the trees, knowing that with those trees, those root systems are going to be able to go down that deep. You just might want to make certain that they're actually going down that far. And so you're going to want to have things that you can, put down a far distance. There was a person at the University of Georgia who was working on that, and I haven't kept up with him. So assuming that I think it's Iowa State University, we sort of challenged them about how deep do the root systems grow in little shrubby things. And so I can't remember the name of the plant. I'm not good at the names of plants, but ask me about bacteria and fungi. So here was the plant that they grew, and they had cut it off at the surface, brought that back to the laboratory, and then started digging. How far down did the root system of that little shrub go? And it was something like 40 feet. And that's just a shrub. How many of us need to be pulling nutrients back from 40 feet down and putting them back onto your property? You don't need to be looking for woody material anymore. Just mine your own soil.
All right, let's see. We have two people, similar interest or question here. Michael says, my orchard is on a plot of land that had the top layer of the soil removed during development. It was a subdivision. So it left behind a rock hard dead dirt. What would you recommend to begin rebuilding the soil, especially vertically? And do I have to build up from the dead subsoil, or can I go down and convert the subsoil?
You will convert the subsoil. That's much the wiser way to do it. I would suggest that he would want to find or make a really good compost that will promote the kinds of plants he wants to have promoted. So there's a little discussion with one of our consultants, or bring a consultant on and walk the land and decide what he wants to do with it. And then we'll help with, okay, this is going to need a compost in the into this growing season, let's put a nice thick layer of compost. And what are the other plants that you want to have in there? Can we plant them at this time? Or do we have to wait until springtime? Probably wait till springtime, but you never know. So come the next spring, we would do the same thing, spread that compost, really good compost. And we would hope that that client would want to learn how to make really good compost so he can, serve to infect the rest of his friends and neighbors coming over to this. Let's keep the nutrients in the soil. Let's keep our water in the soil. Let's eat things that are absolutely the tastiest and most nutritious ever. And so after you have that compost out for 7 to 10 days, you want to take samples from that area, look and see if the biology has improved. So you've got to take 3 samples from your undisturbed, or you haven't put anything on it, compare it to where you have put the biology on. Is it doing you good? And then typically, everything's going to come out looking really nice. Like, another 10 days later, you would go ahead and check and make sure. If there was ever a time or a place where you go, whoa, what happened to the bacteria? What happened to the fungi? Where we used to have really good levels, and what's going on here? And you will determine that your neighbor probably dumped a load of glyphosate right there to be able to prove you wrong. So you got to look out for those kinds of neighbors. And typically, come the end of that first growing season, you probably have brought things up to the ratios that you want to see them. And from then, you're just going to let them grow. There's no reason for you to step in unless there's like a small area where you can see some disease starting to take hold. Then you want to make a compost tea and spray that top and bottom so that all of that area that was even sort of close to those diseases or insect problems or whatever, and you'll deal with those problems. We want you to always being improving the diversity of the microorganisms in your compost pile. The more diversity you have, the more ability to suppress diseases and not have any immune disorders.
So let me ask you this. So thinking about some of our members who might be just hobby growers, who don't have a microscope, aren't quite ready to be able to look at these things. So 2 questions. How will they know a compost that they get or they make is good?
We have a label on it that is bio. Oh, am I going to remember the acronym? Yeah, BioComplete compost or BioComplete soil, or so look for the BioComplete, and that it came through the Soil Food Web School, and through the Soil Food Web Institute Incorporated. We just have a lot of our consultants that are out there. They also make the BioComplete compost. They have to pass certain balances if they're putting it on trees, or if they're putting it on grass, if they're putting it on tomatoes or onions or something like that. We want to make sure that the organisms are there that are supposed to be there. And so we are always out there kind of looking. Surprise, we're here to take a sample of your compost. We can take a look at it right here and now, and everything looks good. Or, it's the opposite is you have a little problem here. And so you're going to have to stop bagging this, and you're going to have to do what's necessary to bring that biology back into that material that you're calling compost. So we try to catch anybody not doing things correctly, we try to catch them very rapidly. It's our reputation, of course. So we get, yes, do it right.
So, one of the underlying messages I got from some of your earlier work a few years ago is that, all compost is not created equal. And if you go buy a bag of compost at your local big box store, it's missing these things, correct?
Yeah, most likely it's missing those things, because how long has it sat in the hot sun out in the drive-in up area parking lot? And it doesn't take long to wipe out all of those really good organisms, and all you've got is dirt. Well, you can bring it back, but it's going to take some effort. So we want to see people treating their compost with respect, if you will. You don't put it in direct sunlight. You don't let it get all toasty. You must have punched air holes in it, in the plastic, so that oxygen is moving through that bag all the time. A lot of people have gone to putting their compost into the refrigerator. Just don't let the temperature go down more than 40 degrees Fahrenheit, because that tends to freeze. Any lower, and you don't have the organisms functioning right away that you would want to see functioning. So take the compost out of its bag, put it in a pile, and let everything warm up. Maybe grab a handful of this other really good pile, something from over here. They have to all been composted for you to be able to continue to call that compost. Even a handful of something that's, this is really good food, let's put that in there. Yeah, but are there diseases on there? Are there unpleasant organisms you don't want to know about? So you have to make sure everything's composted if you're going to add it into a compost pile.
All right, Melissa asks, do you have any opinions on native fungi versus using beneficial fungi from a manufacturer where the fungi may not be native to that soil?
Yeah, we want people to take all of their materials that they're using to make their compost pile from their biome. So what are the limits of the biome? And the U.S. Department of Agriculture, Forestry and Science have some good books on this is the such and such area, and here are the bacteria and the fungi and the protozoa and nematodes. If you're lucky, you'll get the whole food web in there, because we want everyone to utilize those organisms that are normal to that ecosystem, to your biome. If you go to Pennsylvania and grab a handful of Pennsylvania soil and bring it back out to Colorado or Oregon or even California, you'll discover that you have some diseases that you don't, they're not diseases when you're in Pennsylvania, but they are when they get to Oregon. So you're really running on you're probably going to get disease under those conditions. And people just, it's the neighbor down the street had this disease and it showed up in mine. No, I don't think so. Get a microscope, take a look at what you've got, and it's sort of like, whoa, we need to get rid of this as rapidly as possible. So get the organisms from your own biome. My favorite thing to do is to find where's the state parks, where the national parks, because then they're pretty much undisturbed. And all we need for a sample to inoculate your compost pile with is just a pinch of the soil. You don't need much, because once within your compost pile, that set of microorganisms they're probably free to go, because there's nobody in that part of the biome yet. And you're bringing them in, getting them established, and as long as they come from your biome, not a problem.
Jim's asks, where do soil tests fit into this? So the university ag departments have been recommending soil tests, done routinely and, to see results and differences from year to year. How does a standard soil test fit into all of this?
We usually encourage our growers to get, the standard nutrients that they should have, what's nitrogen, phosphorus, sulfur, magnesium, calcium, that whole typical lot. Do they have any evidence of anaerobic conditions happening? And so those two of the, I think I can, I was thinking I had the nutrients, right nutrients. Yeah, so here, like, we want fertility. What would you do, as a, I'm not going to remember his name, but there are quite a few microbiological tests that you can use, not just a microscope. Only using the microscope is so much more detail than just knowing whether your adequate level of nitrate or poor level of nitrate or too high, whatever. So go back through the nutrients. Yeah, there we go. Monitoring the measures of health. That's one of the slides. And so the typical things are in here: compaction, puddling. Those are, how fast is infiltration into your soil. It's all about biology to get that information and get good infiltration into the soil. So yes, there's the typical chemical and biological treatments that we'd want people. So they have a link back to what was going on before, and now they can convert over to just using their eyeballs and using a microscope. And typically the microscopes are, we're not asking for anything really snazzy or very expensive. Now, if you want to be a consultant, you will want to get a microscope that's a little bit more in cost. But as someone looking after your own biology in the soil, a microscope, $300 to $400. You don't have to get the oil immersion lenses. That's not necessary. We know bacteria are small. And so get something that's going to go along with what is easy for you to do.
So we have Lisa, who's in Southern California. She says, in California, everyone is now mandated to compost all organic material, whereas before they were told to trash viral and fungally coated plant material. So what's your thoughts on this?
Should just give us more really good compost. It's the way we should have been living for the last 200 years. And even if you really understood the history of agriculture, it's the way we should have interacted with our local resources 2,500 years ago, when agriculture first really got underway. We've destroyed so much land and just turned it from something hugely beneficial to desert. When do we learn? We have to come back the other way.
So that kind of brings around to, she says, do we need to be more cautious and learn of local trash management procedures using humanure with their inability to filter out all pharmaceuticals and chemotherapy agents?
Yeah, we would want to still kind of keep things that were really extremely detrimental and do very, very careful alterations of that, those, the carbon chains, the proteins, the amino acids and things. We'd want to be really careful of that. But as soon as you've run it with these certain microorganisms in it, those toxic materials are gone. And so you can move that into, now we're going to compost this like a normal set of materials are composted. So as long as it's organic matter, we have organisms that can utilize that. That's, we've had some work with water treatment plants as well, showing them that passage through a compost. As long as you know what's in there that's out of whack, we know how to get rid of it typically.
So that brings, somebody else had asked about plastics in the soil. And I know microplastics has become a huge concern and issue because there's so much stuff made of microplastics and we're shedding it off into the water, into the soil. How's the microorganisms handling that? Are those getting up into plants?
I suspect that Mother Nature is perfectly capable of dealing with this. Plastics and temperature and things like that are very important, but I haven't had the time to really focus down on those situations. So I'm sure if you sent a query into Soil Food Web School, they would be able to go through the school and ask anybody, is there somebody who has expertise in the plastics? And I'm pretty sure we do, but I'm not absolutely sure. So I'm just going to leave it at that. Give us a ring at soilfoodweb.com and we'll let you know.
All right. And Ed Campbell says, what would you recommend a fruit grower to do to change current methods of growing fruit crops? There are many ways, yet I do see commercial growers using chemical sprays into leaf structure and Roundup to prevent weeds. How do you convert?
You just close the top of the toxic material and try to get rid of it as best you can, and start making compost. We've got the recipes. The recipes do depend somewhat on what you're growing, where you are on the planet. If you live in South Africa or do you live in the North America, there are slightly different design requirements for making that compost. And we go through those in the compost, teaching you how to do composting. So yeah, it's really not difficult at all.
All right. Someone asks, what mix of cover crops would you suggest for fruit orchards to optimally feed the soil microbes in Zone 9 of California?
Zone 9 is up in the northeast part of California. I don't have the zones of California state memorized. So what you'd want to do probably is go looking for some books that talk about short, low growing types of plants, so that the cover plant that you're growing isn't going to be influencing or be a problem for the crop that you desire. So make sure that your cover plant is shorter than what your crop plant is going to be. So if you're growing trees, I don't think you really have to worry much, other than don't stick in another tree. Bushes, shrubs, all kinds of things, grasses. Just make sure, like with a grass, that it's a grass that's short. People keep putting in grasses that are 6 feet high, 12 feet tall, and then you have no choice, but you've got to come in with your equipment and mow that. Well, why put something in that's so tall if you're going to have to compact your soil in order to put that material on the soil surface? Or maybe you have such a huge amount of that grass that grows that you've got to put the excess someplace else. Well, you could make more compost with it. So, yeah, it's have a conversation with one of our consultants, or get one of the consultants to be your go-to person all the time. And then you can have the fun of working out some of these interactions. What are the best plants as cover plants? And another good place to go looking is in the native plants. It's, parts of places like the university. They have people who look at cover plants, and they have written up significant information about what this one does and what that one does, or what the other. So native plant societies, same thing. They all have information for you.
Great. All right, we're going to give you one last question here. There's still lots of questions, so I know we're not going to get to all of them, but for the hobby grower, again, we have a lot of hobby growers, and we kind of talked about this a little bit. How can they find out about the bacteria and fungi ratio in their soils? And someone suggested a microbiometer testing kit for $200. Do you recommend that?
No, and they don't give you the kind of information you want. They give you DNA analysis, and I haven't seen a lot of where those two things overlap at all. So for, you want to just be looking for things that you would be growing anyway, perhaps. Go to all different kinds of stores and ask them what grows no taller than this. Figure out what that height should be for your plant, for the above ground. I feel like I didn't quite answer this all together. Did I get everything, or did I miss something?
I think, from what I can tell, it's hard, to be able to determine the bacterial and fungi ratio is difficult, but I think you can look, correct me if I'm wrong, but you can look to see what's growing. So if you're getting lots of weeds, then you got a lot of bacteria, right?
Yep, and you need to move along to the fungal. And it's very easy with a simple microscope to take a look at what's in your soil. It takes, 5-10 minutes for you to look at a field and go, all I'm seeing are bacteria. This is not good. This is not going to be selecting for the growth of my crop, much less a cover crop. And so that's how you tell. Anytime you have a question about what's going on in your soil, you take a sample, you look at it, and you find out what's going on. And if you don't know completely how to interpret things, again, that's where you're going to be pulling in me, or you're going to pull in somebody else that you've been working with on this, on getting the right crops and the right cover plants into the system.
So ideally, in the years to come, we would have every extension agency have someone on their staff who could do the microscopy for their farmers and their growers.
Mm-hmm. And we do a lot of work where we look at C to N ratios and different situations. So we have books that have those kinds of relationships. And so if you come and take the foundation courses, you'll get one of those books that kind of go on and on and on with what's the C to N ratio. And then we're going to show you some of the statistics. How variable was this? And in some plants, it's really important to differentiate between a certain kind of this kind of plant, closely related in terms of DNA structure, or what media are they going to grow best in, and you'll discover they're extremely wildly different, and you have to pay attention when you're seeing some of this versus some of that.
And so we had one person who, John, who just pointed out, and you've mentioned this, but just to hit this home again. So Dr. Elaine's website, you can go there and you can find a soil food web consultant or someone to send your sample to who can send you back a result. So she's got people trained on the microscopy. You can just send them your soil sample just like you would to your ag extension agency. Yep. And they can come back with you and tell you how much, what your ratio of bacteria to fungi are. So that's another great way of doing that. So Dr. Elaine, I want to really thank you for spending the time today to go through this. It's a lot of information for people to take in and absorb, but I highly recommend people to go to the website, the Soil Food website. There's also this video that will be made available to all of you, and she's got lots of other videos. If you search YouTube, she's out there everywhere. I first heard Dr. Elaine through a podcast, and I was just amazed at all the great information. So just take it in. It's a lot to take in, but you know, the more you listen and the more you read and the more you take it in, the more you'll understand. So on behalf of NAFEX, I'd like to thank Dr. Elaine for making this a wonderful session. And we also had sponsors this year, which helped make this conference available. So One Green World was our major sponsor, Copenhagen Farms Nursery, Fedco Seeds, orchardpeople.com, Southern Cultured Orchard and Nursery, and a couple more sponsors. Once again, this session was recorded. It's going to be made available to NAFEX members through our website. You'll log in to access that in a few days, and we'll send out an email to let you know it's ready. And then after that, we migrate it to the NAFEX TV YouTube channel. So past conferences are already on that YouTube channel if you're interested in those. There will be downloadable content. Many of the speakers are providing their PDFs, so you can go back and review material if you'd like. So that will be available on the website as well when you log in. And please stay tuned for what we plan next year for our conference. We talked about this a little earlier today. It takes, you know, a village to make this happen. So lots of volunteers. We're a completely 100% volunteer-run organization. So the more the merrier who can help make this possible. So thank you again, Dr. Elaine, for presenting our keynote today, and thank you all for joining us. And you know, send us your feedback. We'd love to hear from you. And you guys have a great weekend, and we will see you at some future online meetings.
Can I do a quick explanation of the picture? Oh, you're probably not seeing this, so never mind. I was hoping I could get one picture of here's with biology and here's without, and the chart is growing this tall with the biology, and right next to it the part of the field that didn't have any biology applied is only that tall, and they were all seeded on the same day.
Oh, that's awesome. Is that picture available on your website?
I believe so. Yep.
Yeah.
So come and take a look at
You know, what really strikes me is I've seen it in some of your videos and some of your coursework, but the before and after and the side-by-side fields are just phenomenal. So if you guys can find that and check that out, definitely worth your while, because it really solidifies how just a little bit of input in the bacteria and the biology can make a huge difference. So thank you all. You guys have a great weekend, and thank you, Dr. Elaine.
Okay, thank you for inviting me. This has been fun.
Good. Bye.